Can you hear me well?
Yes.
I want to apologize ahead of time. My voice is MIA tonight a little bit, so I will try to do my best. We also had some technical difficulties, so I will have to stick around here to advance the slides. This is a vast topic, so I'm going to go on a quick survey tour in three parts. One is just a general introduction into the subject. Two is just a brief overview of the tools that we use and that are available out there. And three, I will tell you about two projects that I'm currently involved in, so it will be relevant to what we're talking about. You can see what we're doing right now, so kind of tales from the front line. Can someone tell me what happened in, considering that we're talking about bioinformatics, what happened in 1953?
DNA.
Yeah? Great. So this is the famous Watson and Crick 53 Nature paper. It's just a page, but it opened the door to a new kind of science. So we are accounting for the bio. What happened in 48 that accounts for the informatics? Anyone heard of Claude Shannon? Okay, so this is the other paper that is a foundation for bioinformatics and many other disciplines, a mathematical theory of communication. This is actually the father, well, he's widely considered the father of information theory. We're only briefly going to touch on what information is and how it's measured because it's another vast field and we don't have time. But let's go through several definitions of the field. This is Wikipedia nowadays, so you can't give a presentation without citing at least one. So it is the usual interdisciplinary scientific field that's for storing, retrieving, organizing, and analyzing biological information. When did bioinformatics appear as a term at all? So somewhere in the mid-'80s, and in the beginning it was very general. It was everything from robotics to artificial intelligence. Afterwards, people saw that the computers were not fast enough, so they couldn't do all that stuff. So they toned it down a little bit, but then it became a little too prosaic. It's just applying computational, using computers to work on biology. Now, and then the narrow definition that still lingers around these days is that it's the application of information technology to the management of biological data. Now, this is too long, but this is one book that I'll recommend for anyone who is interested in the subject to pick it up. It is kind of medium level, upper undergraduate. So what it says is that all these definitions miss the point. Information is fundamental to life, whatever we define as life. We'll talk about that a little bit. And therefore, bioinformatics can actually is now a mature enough science that it can pick up the quest for the answer to the question, what is life? And this is a much more deep definition. It's not just using computers to crunch data that comes from biological systems of some sort. It is actually, it has a goal that is still unreachable, but it informs a much deeper type of research. So, I'm not going to answer this question, but if you ask yourself this during the lecture, it will help give us a little bit of perspective. And what does life have to do with information? Let's start with information first. What is information? That's also a very difficult definition, but here's an intuition. If we lived in a world that only had one type of object, a ball, and they're all red, there's no variety there, would we even talk about information? What information can we transmit that will enrich our view of the world? It's just balls and they're all red. You can't ask me a yes or no question that will give you any more information than you already have, right? Okay, so how about variety? If we have many colored balls, then you have to ask me several questions to if I'm holding a ball, you can start asking me, is it red? And I'll tell you yes or no, so on and so forth. Now, variety creates a world in which information can decrease your uncertainty about the ball I'm holding or anything else. So information is a measure of your decrease of uncertainty. It depends on prior knowledge. If you knew that if you hit all the balls that are green and blue, and I'm obviously holding a red ball and you don't have to ask me questions, so information depends on your prior knowledge. Now, there's a very sophisticated mathematical theory, you can read that paper, about all these notions about variety and constraints and prior knowledge and so on and so forth, but it is a little beyond the scope. But this intuition should take us a long way. And information, most importantly, can be quantified. I can tell you that for you to find out what ball I'm holding, I will have to transmit 1.59 bits of information to you. So, what does life have to do with information? Watson and Crick opened the door of a view of life that is a lot more intricate and a lot more about passing on information from generation to generation. And this is reflected in what's called the central dogma of biology, or bioinformatics also. It is in commas, we'll see why. Nothing is so clean in biology. Is everyone familiar with what's on the yes, no? So DNA, RNA, and proteins are macromolecules, polymers that are in the very heart of what life is on Earth. Maybe there's other types of life out there, we don't know. But organic life on Earth is all about DNA, RNA, and protein. DNA holds the information, more or less static. Actually, that's skirting around many issues. RNA is a transporter. It's a messenger. It takes the information to the machinery of the cell. And proteins are the products. So this is very clean and nice. What are proteins? This is a famous poster from one of the best illustrators of proteins. Don't expect to ever see them like this in color or anything this high resolution in real life because they're too small. So you cannot see wavelengths that go that far. Really, proteins are everything. They are the machinery of life. They have multiple functions. They are, in the human body, there are about 20,000 different proteins, and they do so many different things we have only started to scratch the surface. This is just an overview. You can see they vary in size. Somewhere here is a water molecule. It's right here. You can see it. This little dot is a water molecule that shows you how much bigger these macromolecules are. So, because of this central dogma and all this replication of information and this passing on of information and then acting on the information to produce products, post-genomic biology has become an information science. There's so much encoded in the DNA and in the processes of RNA, the transcription, and in the protein-protein interactions or interactions with proteins and other molecules, that the only way to manage all that information is to resort to information science. So I'll just run through a very quick overview of how we present biological information. As you can see, this is, what is this? Can you see that? This is basically a gene of a virus, and these are the different regions that code for different proteins that are involved in its life cycle. You know, viruses defy our definition of what is life because they're kind of halfway in the middle. They can infect us, but they cannot reproduce without a host. So, proteins. Proteins are sequences of amino acids, but they have several layers of structure. And that structure informs their function and their interactions, and there's so many different levels of studying proteins. Primary is just the sequence. Secondary is these highly preserved elements, alpha helix and beta sheets. Tertiary structure is where is studying the actual final fold of the protein. Proteins fold in on themselves to make globular objects. If you remember this here, they're all folded. And that fold is extremely important. If it misfolds, you get cancer or a lot of other things. Representation. So proteins have subdomains, domains, different regions that are responsible for different functions. And this is one way of representing it. So, these little regions do different things. They bind here, they catalyze, they attach, they detach, and so on and so forth. Very often, proteins, when they interact with others, other elements in the cell, they change how they're folded, so it only complicates things. So there's also a full atom-level representation, simulation of what's happening on the biophysical level. So this is a protein that actually crosses the cell membrane and has some transporter function or a signaling function. These are very expensive, as you can imagine. Every atom has to be represented the way it would act on a quantum level. Now, bioinformatics doesn't just take one protein and try to understand it because they're not very easily directly observable. So what do? We aggregate the data across a lot of sources, a lot of species, a lot of different microenvironments, and we compare these sequences and what's in this one that isn't there, and so in the other one, and we try to infer function from that structure. So this is the human genome as in its chromosome form. If you were here for the epigenetics lecture of Dr. Phil, you will know a lot more than this right now. But this is a very complex fold of the information, and there are many other non-genetic changes that can happen there because of this organization. What is this? These are genes. This is DNA. Right? This is a very popular way to find out what proteins you have in a solution. You take it from some microenvironment and you want to see what's there. So this is called a proteome. A proteome is a set of proteins that are taken from somewhere in particular. They could be the blood plasma proteome, that all the proteins that are moving in and out of your veins and capillaries while you're alive. That changes minute to minute. So this is a static snapshot of that some microenvironment, and it's a 2D gel. One of the dimensions is mass, the other is charge, and they can be identified. Strangely enough, it looks like just a lot of dots, but people know what they're doing. We talked about DNA and proteins. Remember, in the middle there was RNA. All the transcription products, there are all kinds of RNAs. There are longer ones, smaller ones. They have very many interesting functions that are beyond the scope. But this is how people study what's in there. So these are microarrays, you might have heard. So they're little plates with that are prepared in a special way. You put your solution there, just a little drop in each one of these wells, these little wells, and depending on what is bound there, they're also phosphorescent. This information can basically be taken out and studied in a network comparative way with others and so on and so forth. We'll see how data is collected, but this is basically a very one of the many ways. I mentioned that proteins interact with proteins. The full network of protein interactions is called the interactome. As you can see, you can't see much, but what I can tell you is that our most optimistic estimate is that we know about 12% of the interaction network. So these might be all the proteins, but about 80% of the connections that should be there are not yet. We don't know. So life has to feed itself and has to metabolize energy all the time. The network of those metabolic reactions and pathways is called the metabolism. As you can see, it's huge. You need a 72-inch 5M screen to just see it. And the final one that I'm going to mention here is that we're not alone even in our own bodies. There are a lot of bacteria, and I'll tell you how much and what that might mean. We're only starting to understand what that population that's called the microbiome, how it affects our being, our health, our many things. There are two projects that you can join that are crowdsourced. They send you a kit, you give them samples from all your orifices, and they sequence your microbiome. And they give you that information, and it can also end up in other people's hands, but it's an interesting kind of way to participate on the forefront of science. So let me talk a little bit about beyond the dogma. So I put the dogma in quotes because the picture is a lot less clean. So there's alternative splicing. The RNA that copies the gene might undergo spontaneous or induced rearrangements. So you can imagine the protein is going to be quite different if it's functional at all. I'm just going to mention all these because there's a lot of science that's behind. Post-transcriptional regulation. I mentioned there are a lot of types of RNA. You might have heard one of the more interesting ones in the past few years is the RNAi, which is an interfering RNA. So this is a small sequence of RNA that can block the transcription that is already in progress, the expression, the production of a protein or something else. Non-transcriptional expression regulation. So proteins are involved in copying the DNA. They're promoters, they're inhibitors, they're transcription factors. They attach the DNA to the proper places, and if they don't, that protein that is downstream on the gene doesn't get produced, or if they're promoters, that they produce it in more abundantly. The interesting thing is that our behavior can be so some of these expression regulation processes can be traced down all the way down from our macroscopic behavior, the way we live, the way we eat, the way we live our lives. That can actually affect what happens in our bodies. So next time you're in Zen country, give them the benefit of the doubt. Epigenetics, we talked a lot about that. Epigenetics has to do, in a previous lecture obviously, has to do a lot with heritable but non-genomic changes. So non-nucleotide changes. They're not changes in the sequence, your DNA sequence, but they're changes in the chromosomic structure of your genome that make it easier or more difficult to express, and that can be inherited through sexual reproduction. And then I mentioned the bacteria because they don't have they have a process that we don't have. So they are prokaryotes, where we are eukaryotes. These are two different cells. They don't have a nucleus in their cells. We do, but that's only the beginning of the difference. One of the more interesting ones is that they bypass the machinery of the ribosomes, the transcription, and make peptides, which are short proteins, directly from the DNA. And they live in our bodies, so that affects us. So, what about these guys? So, look at that. If the human genome has G number of base pairs, then the collective genome of the microbiome, these are all the different types, not the different cells, but the different types of bacterial cells, have 10 times that in terms of genetic information. In terms of number of cells, they're 100 times more than our cells. They're much smaller, obviously. You don't have outgrowths of bacteria, but they're there. A little bit, I'm continuing my Beyond the Dogma elaboration here. At this point, we've only accounted for about the function of the 50% of our genome. 50% is still out there. We don't know we know what it is because we got ourselves sequenced, but we don't know what it's doing. And You've probably heard about genotype versus phenotype. Genotype is what information is coded in your genome. Phenotype is what is being expressed. This is the interaction of the genome in the environment, and this is you. Now, think about this. 98% of the genome of man and ape is the same, but they're totally different animals. So there's so much that we don't know. There's so much implicit information in the organism that is not coded in the genome, in the DNA, that is yet extremely influential to our lives. I don't know if I told you you'll never see the cell like this, but after a lot of research, people have actually managed to draw it. What's interesting here is that our ideas of the environment, of the microbiological environment of the cell, are so and we're guided by our intuition, by our macro world. Usually chemical reactions are done in a test tube in a lot of solvent, low concentrations of the reagents. Here, everything is packed. Proteins are rubbing against each other. They are changing their folds all the time. And so this looks a lot more like a micro machine than a chemical reaction test tube. And many other things. So we're only starting to understand the intricacies of this, of what's happening. So what do? We model. Science is basically the best current guess of what we understand. So biology is turning out to be the most complex science that we've ever tackled, and it's becoming more and more complex by the day. Bioinformatics is taking a bird eyes view of biology and trying to understand it as a system. So it's putting all the information that we've accumulated over decades of research and it's putting it together into models. So I'll just fly through these. There's several different approaches. Systems biology is sort of self-explanatory. Systems are complex entities that have a lot of machinery underneath. Another way, metabolic pathways. So I showed you a metabolome. This one is even bigger. Complex systems. The idea of complex systems is that small elements of the system with very simple behavior, let's say proteins, combine to create very complex emergent behavior on the system level. So we're trying to study that as well. Biology hasn't become quantitative yet. There's so many, as I said, the machinery is so intricate that we are finding it hard to model it down to the concentration, down to the number of atoms, and it is very. You'll probably hear this word, so I'm just going to define it here: stochastic or probabilistic, non-deterministic, basically. If a protein has an option to react or not to react, there is a probability that it will and a probability that it won't. And these probabilities change depending on the environment. So it's complex. Wrapping up here. So all these discoveries, all this information that we're amassing, eventually it has to drive something more than science for science sake. So translational bioinformatics is what takes this from the lab, from the bench, to the bedside, to the clinic. It is a very intricate socio-economic, scientific problem that is beyond the scope, but if you're interested, I can talk about that. I want to wrap up that bioinformatics has a lot of sister disciplines. There are no clear boundaries. This is a fact cloud here from the Bioinformatics Wikipedia page. And you can see there's systems biology there, there's computational biology there, if you just put them together. And they're all different fields, but they're doing similar things from different perspectives. So, I'll stop for questions, and I'll probably only have time for the second of 3 sections, but I've talked a lot. Let me know does this make sense? Do you have questions? Yes?
Well, what kind of functions do the non-ribosomal peptides have?
Peptides have a lot of functions. I can't even begin to it. So they can regulate reactions. They can be consumed as metabolites, and these are only 2 large. They can be used in signaling pathways as well. Many different. So they are just like proteins, but they're shorter. Peptide doesn't have a very interesting fold. It's usually just a ribbon, because it's up to 15 amino acids. That's all.
I'm happy to come to you. Thank you.
Anyone else?
What would you consider the current major choke point for bioinformatic research at this point? Is it the information systems? Is there enough scientists? Is there enough money? What would you consider the problems right now?
The biggest problem is that we don't know what to do with all this data. Let me put it this in a different way. The information that we've amassed has surpassed in volume and intricacy the capacity of a single human mind to comprehend it. So we have to resort to computers and try to see sections, aggregations, patterns that we can see in big data, but we cannot process that by ourselves. So we crunch these numbers, we visualize, and we look for regularities. This is what science is, look for regularities. But the information is a lot. It's coming at an exponential rate. That means that every day 112 articles are being indexed in PubMed. This is the biggest database of, it has several tens of millions of biomedical articles. Who can read these? You cannot read 112 new articles. Let's say that you're only interested in the ones in your field. There may be 10 a day. What do you do? So we're going to try to mine that literature and maybe change the way we publish so that all that information goes into the models that we've already created and triages them and improves them and so on and so forth. So that's a big problem, and it's not going away, unfortunately.
All right, maybe some.
Okay. So, short survey of the tools. Bioinformatics is now mature and accessible. Every new field starts with very few available tools and very high, low level of accessibility or a high barrier to entry. Usually pioneering scientists do the work, and then expands to graduate students. That's nice. We have a lot more of those. And now bioinformatics has reached the place where there's a lot of work for even undergraduate students. That doesn't mean that everything in biology and bioinformatics is already discovered. Not at all. You saw, if you've seen the epigenetics lecture of Dr. Field, he's up there. Epigenetics is right here. I'm going to talk briefly about data structures and algorithms since we're talking about informatics. So, data structures. sequences are abundant in biology. We have DNA, RNA, proteins, and many others. They can be represented as strings, like this. So this is an attempt to match two different portions of genome of two different organisms. Or as trees. We know about trees, right? They have roots, and they have branches, and they have leaves. This is technically not a tree, but we're not going to go into that. So trees don't have cycles, right? You don't see, well, there are some tropical trees that have cycles. Another data structure, networks, the graph. So a lot of networks, as we said, interaction, the interactome is only one. So this is an interactome of a virus and some proteins involved in cancer. Patterns. Do you see the pattern? Do you see the dog? Okay. Patterns I've attempted to define them, but everybody understands there are so many different fields that deal with patterns. They are structure co-occurrence of some features that are interesting to you. So this is a pattern, this is a dog. Well, it's not a dog, it's just a bunch of dots, but you understand it as a dog. So pattern is what you do with it, and there's pattern theory, and that's a very deep field that I'm not going to go into, but looking for patterns, that is the regularities in science, in bioinformatics, this was one of the foremost things that we did. Pathways. Pathways are sets of interactions between proteins that have a certain common function. So these are not just interactions, they're actually signaling a way of for the cell, the inside of the cell, to receive signals from the environment. Why does it need signals? Well, it has to coordinate with so many other cells in your body. You've noticed that your organs do something they have a purpose, right? That purpose means that all these constituent cells are doing something together. That requires a lot of information passing. So these signaling networks, these signaling pathways, is how it's done. These are, by the way, proteins. Obviously, they're just little blobs, but they do have that intricate shape that I showed you earlier. Back to the modeling on the atomic level. This requires multidimensional data structures. It's not just data structures, it's full models. So there's actually a lot of functionality in that data structure that is active during the simulation. Algorithms. An algorithm is basically a bunch of step-by-step instruction of what to do with the data structure and how to get the result. There are two interesting things about algorithms. How much memory they take to do their work, and how long they take to give you a result. This is the simplest. These are the simplest criteria for complexity. They're a function of what you give it. The algorithm takes data structure, as I said, that has some information inside. Depending on how big that data structure is, how big the tree, how big the genome, the memory and the time consumption is going to be obviously different. So it's a function of the size of the input. Does that make sense? Sometimes it's not just the size, but also the contents. So depending on what the contents of of the data structure, you might have a variation of resource consumption. And you should, if you expect that such big variation, you should always compute your worst case scenario and your average case scenario, and how often you get into a worst case scenario. That you can see that from the regularities in your data. I'm going to skip this because it's a notation that people that deal with computation use to approximate complexity. I'm just going to say that something that has linear complexity is linear in the size of the input. It's very nice because, you know, if you give it 10 times more data, it's just going to take 10 times more time. If it's exponential, though, if you give it 10 times more data, it's going to be 2 to the 10 times more time. Eventually, a lot of those algorithms get so complex or so big, the input is so big, that they will never finish in your lifetime, not in your lifetime, but in your lifetime of our planet. So what do? We approximate. We cut corners. We don't want exact solutions. We go for approximate solutions. And we use heuristics. Heuristics are some guiding principles that we use in an algorithm to basically show it which approximate solutions are better than other approximate solutions. But very often we don't know what the best solution is or whether it's attainable. The final thing I want to say, the constraint, so this is bioinformatics, it's not just informatics. The result of the algorithms has to make biological sense. Very often you find an awesome new solution to a problem, an algorithm, you get a result that absolutely doesn't match the prior knowledge about the biological system that you use. So you have to go back and iterate. Data. As I said, the biggest problem is that we have way too much data. Well, this is also good because we haven't had it before, and now we can look for. There was a saying in physics in the beginning of last century, of the 20th century, that the most interesting discoveries are made, 6 positions after the decimal point. So that means that you have to have very sensitive apparatus to make your experiments to find out some weak interactions that actually are very important. With this amount of data, we can actually have a very high discriminatory power because we can see what is. We have a much more background to see, oh, okay, this is orange, but now if you put it here, it's actually more like red. This is a huge topic, data versus information versus knowledge. Just have it in mind. It's very interesting. How do we collect data? High throughput assays. So these are robots that deal with these arrays. For the transcriptome, I showed you an array like this. So you do it very fast. They do it for a lot of organisms, so for a lot of samples, and all this data basically, is put in a database, and people start bashing at it. The exponential growth. So this is the number of submissions to the European Bioinformatics Institute for short reads, are basically small snippets of DNA that people have found somewhere, and they want to see, well, what does that match in what genome? Look at that. We're talking. What do we do with that information? A lot of the information is actually being organized in publicly curated databases. As of 2012, there were 1,380. One other way that we try to structure our knowledge. If we go back to here, data, information, knowledge. This gets going from the left to the right, it gets a lot more structured. Data, which is numbers. Information, we know what the value is, what information, what extra information we're getting. Knowledge is a digestion of the information. Sometimes, I draw you a picture from my knowledge that is down to the atomic level, and you get the general idea. So that's knowledge. Ontologies. Ontology. The term ontology has two different meanings. One has to do with with creation, with how the world came around. The other is a structured representation of hierarchical knowledge. So look at that. Mouse anatomy. Adult mouse organ system. Integumental system. So this is a type of system. Then we have hair follicle. You see, it's a hierarchical representation of the knowledge. There are a lot of uses for ontologies. They're used to annotate data sets. So this, if we go all the way back to where I told you that polygenomic information, polygenomic biology is an information science. All these things are annotations. These are IDs for proteins in certain, or genes in a certain database, and there are a lot of use for ontologies with exactly this annotation. Eventually, you get from the information, you get knowledge because it's already structured. And if you start annotating your data, you're like, okay, this actually shows this biological process. Sorry about this. And knowledge bases. These are very heterogeneous collections of information and knowledge. They can be constructed as databases or just repositories, and there are many different types. There's a lot of information in these 112 new papers every day, but who can read that? Computers cannot. So we just throw away all that information. What happens in science is that you publish, and 5 days later you cannot remember your abstract. 2 years later, you cannot remember what you wrote about. And this is because you move on. We can only focus on a little portion of what we are doing, and we move along the discovery line. But all that information gets shelved, but we want to use it. So, we're using natural language processing. So we're doing very deep analysis of text, and we try to find regularities. People are very good at natural language. Computers are very bad at natural language. Natural language has a lot of variety, a lot of irregularities, a lot of ambiguity. The same word means totally different things depending on the context. The context is a lot of data to go through to understand. We are born with that, and our nervous system is a hierarchical representation of all that world knowledge that we have. But a computer doesn't have that already. So it doesn't ring a bell here and there. So the sense, the meaning has to be extracted from a lot bigger bodies of text. And so you see the scale of the problem that we're trying to. So what we do if we read the text, we actually turn it into a more regular representation. Called them triples, basically. We have a subject, an object, and a property. In my lab, three of my colleagues have created one of the biggest in the world repositories of integrated information that has a common biological model. So all these different databases that are 35 databases and 12 ontologies, they've actually made them correspond to each other. And we have 10 billion triplets. Just last year, there was a world challenge for being able to handle 1 billion triplets. These are just basically, think of them as simple sentences. Okay, I have 5 minutes for questions, or I can run through 2 projects that I'm working on. I'll fly through them, but let me see if you guys have questions and have to catch a bus or whatever. Projects. Okay. So this one is called Protein. Remember the protein? Protein is a set of proteins, right? So this is my little company, which is 3 of us. So the reality of drug design. So it's a commercial entity, so it has to sell something, right? It takes, on average, $9.6 billion and 12 to 16 years to develop one new biological drug. Oh, I lost the color here. I'm sorry. So this is a very short protein, just 50 amino acids. These are a lot of variants. What does protein engineering do? It makes changes. Let me find a change. Here, L and V. So this is a single substitution of an amino acid, and we do these substitutions and look for the best variants of some protein for our purpose. It could be a drug, it could be a drug delivery system, it could be a bio catalyst to make biofuels. It could be anything. There's a big problem with it. These amino acids have a common backbone molecules, and they have all these different radicals. Look at that. They're so different. So when you make a single change, your parameter, you can actually completely change the protein. It might actually break apart. It doesn't hold together anymore, or whatever. This is how the space looks. So there's no regularity to it. It's very hard, and this is a huge space. Imagine each one of these places. So these are 50 places. Each one can have 20 different. So this is 20 to the power of 50 different variants. There's no way in hell, that we can actually go through all of them and simulate them and see which the best matches. So talking about approximate solutions here. So this is what we do. So this is, for example, an enzyme, and this is an enzyme inhibitor. They have to bind. So we're looking for the best binding score. This is actually a database taken from a database of experimental data that is done for several of these complexes that we use for machine learning. Because, as I said, we cannot do this in our heads. Machines have to do it. These are sampling of the parameters that we need to optimize for a single protein. Machine learning. We actually developed an algorithm that uses information on the biophysical level to improve upon the state of the art from last year. As you can see, these red dots are predictions. This is the data that we have. These are predictions. And our predictions are closer to the diagonal, which is a very good criterion for improvement. So this project is actually trying to use the human mind, that pattern recognition that I mentioned, in an explicit way. So we're creating a game for people to play with proteins, with representation of proteins. Representation of proteins obviously not going to give you tubes to put in your coffee. But so players solve puzzles. They employ their pattern recognition skills. So this is a mobile device. These are elements of the gamification. Hey, you did well. Do you know what your friend is doing? You know, games. Very sophisticated computation, machine learning, as I mentioned, and aggregate all this new knowledge, all this exploration. People don't explore randomly. They go after patterns, something that they learn. Hey, if this is folded like that, then I remember this makes it more stable. There's actually a whole game called Fold It that does precisely that, tries to fold proteins. And people are folding proteins the way nature is. This is folded. This is a turn. Notice the RNA in the suffix. So you build RNAs and they build them and see if they work. This one is called IY. This is a gamification of tracing down the neural connectome of a mouse. So how the neurons of the mouse in the mouse brain are. What do we want to achieve with that? Personalized medicine. As I said in the beginning, it takes so much money and so much time to develop one drug. If we don't all together respond the same way to the drug because we have different genes and we have different phenotypes, so some are going to have a good effect, some no effect at all. Actually, most of the time there's no effect, and some are going to have adverse effects. So this is the way drug design works today. It's called the blockbuster model. Everybody wants to discover the new aspirin that it does everything. It cures everything, but it doesn't have side effects, right? So personalized medicine is the idea that we're going to tune the drug to your genotype. Imagine how much faster we have to produce drugs for you to benefit from that. Second one, very different. It's called spinal cord injury and regeneration. As you know, acute spinal cord injuries leave you paralyzed. As you can see, depending on where it is, you can be fully paralyzed, partially paralyzed, and so on and so forth. There are about 2.4 million people in the world, mostly in their prime, that are paralyzed for life. This is a schematic of what happens in the spinal cord. This is the injury, this is the chronic stage, and then these are possible treatment options. For 100 years, we haven't been able to treat a single case. So we're getting proteomic data to study from the site of the, that didn't work. So we're getting data, samples from these tissues at different intervals after the contusion, the injury, to see what happens. Differential expression you might hear. This is one of the most used methods in bioinformatics. You take a control group that doesn't have an injury and compare the proteins that exist at 1 hour, 3 hours, 6 hours, 1 day, and so on and so forth, in the injured tissue relative to that. And you start getting an idea of what's going on there. You might. How do we get this? From mass spectroscopy data. It looks like this. Basically, these are proteins with a lot of with relative concentrations here, and some of the annotation from the gene ontology of what they do, where you can find them, where in the cell, what biological process and what function they might have. This is a sample of the protein-protein interactions between because I just picked one of them and look how many different interesting possible interactions it's going through. This is the data. On the literature side, there are numerous, so we make a search on spinal cord injury and spinal cord regeneration, and we got all these papers. So there's a lot of information, extra information about these processes there that we might actually get out and add to our model. So this is the thing that we do. And of course we are creating an ontologies specifically for the this is something of the geontology. What I'm saying is that right now we are creating our own ontology to be able to have a hierarchical understanding of the process and the mechanisms. Eventually, all these interactions are going to make their way into a model of the pathways, different pathways. These are more structure on top of the protein-protein interactions and more knowledge about exactly what is happening. And we correlate that with the differences in the differential expression. We might actually get clues as to what we're not, what we're missing. And this is one of the early papers. This is our Barbara Grimpe in Germany is our principal investigator. They found that one of the reasons for for the failure, regeneration failure of the spinal cord is that the extracellular matrix proteins, the proteins that hold the cells together, which is a very complex system. One of these proteins is overexpressed. That is, there's a lot more in the injured tissue. So they reduced that, and they measured. There was a measurable improvement of the success of neurons, of axons, trying to get past the zone of the injury. So we get some good results. I think this is it. Thanks very much. Take questions? Yes.
So on the spinal cord one, are you comparing spinal cords that regenerated or healed to those that are not? And are you seeing differences there?
No, the control group is non-injured spinal cords.
Okay. But some spinal cord injuries can be healed sometimes soon after.
Yes.
Speak into the mic. Okay. So if some can be healed, is it worth looking at those? Because that's
Some can be healed, yes. We are looking at acute spinal cord injury. So that does, the maximum damage. What can we do about those? Because there are partial, there are a lot of cases of partial injury, partial paralysis, or not such severe effect that can actually be healed normally, because there's no the injury here isn't that bad. So these are different cases. We believe that acute spinal cord injury, and we have a specific model of that. Has different mechanisms in which it forbids the regeneration than these partial injuries. See here, partial. We basically work in this area here, complete injury rather than partial injuries.
So the hope is eventually.
The hope is eventually we can cure people of paralysis, but there'll be a lot of years before that is done.
But even if you know the protein is overexpressed, is there a way you can tone it down?
Yes. That's what this paper is showing. I'm sorry. if we artificially reduce the expression of this protein in that tissue, the neurons will try to regenerate and will be more successful. But that's only one part of the mechanism of the complex mechanisms that we are expecting.
So this has been informative for me with the ontologies and the data processing and access that you're talking about. Does that translate to the NSA and their information system? Accessing conversations, emails. They have a similar problem, I would presume.
They have a similar problem, but they have different prerogatives. And they have a superior budget. What else can I say? I don't want to offend anybody, but that's the truth.
Even let me have a question. You mentioned that sometimes you get results that are not consistent or compatible with knowledge, general knowledge we have in
Right.
biology. How often have we found out that we had completely wrong concepts, or how often do we really have to go back and say something is wrong with the way we approach it?
Well, as you heard in my introduction, I'm not a biologist, so for me it happens more often.
I'm curious, as to maybe privatizations, in this industry, that kind of thing, where.
Can you hold it closer?
Yes, privatization, and say two companies going after the same target, maybe 6 companies going after the same target.
Yes, it happens a lot.
So is there some kind of maybe way to get people to solicit their data,
Get them
share the information,
all to work together.
and then maybe have a residual effect when somebody looks up their data, or has, if we donate it to the pool and we're not all competing. Is there any project like that would
Yes,
solicit data?
because the old way of doing medicine has failed, is failing. People looking for new models, and there are a lot of interesting business models that try to add a lot more collaboration. As you can imagine, this culture of secrecy, patents and everything, lawyers and everything. A lot of the information is not published, so you don't know what the others are doing, and there's a lot of duplication of effort. So a lot of money is going for the same thing, and if that thing is a failure, then 6 companies are failing simultaneously. Great. Who's paying for that? So there are there is the new horizons, but they're still on the horizon. It's still about blockbuster and big money, but they are trying. They don't have a choice. Biology has trumped that. The complexity of biology has trumped that simplistic view of how we respond to medications and treatments. It's a lot more complex. So we just have to play along. Thank you very much.
Thanks.